Hello, thank you all for joining us for another great session at the Baird Healthcare Conference. My name is Jack Allen. I'm a senior biotech analyst here, covering the cell and gene therapy space and also kind of the regenerative medicine space, which brings us to our next company here, Frequency Therapeutics. We're joined by the broader team. We've got the CEO making a presentation, David Lucchino, and I'm going to hand it over to him, and then we'll have some Q&A with the additional team members as well. Thank you so much for taking the time to join us.
Thank you, Jack. Good afternoon, everybody. My name is David Lucchino. I'm the CEO of Frequency Therapeutics. I'm joined by my colleagues, Kevin Frank. Kevin joined us about a year and a half ago from Harvard Medical School, where he led all of audiology. And then Chris Luce, our CSO, who is one of the co-founders of the company. The obligatory forward-looking statements and other disclaimers. Please let me know if you have any questions. So 2023 is the year of the clinic for Frequency. In Q1 of 2023, we're going to have a readout from our phase 2b 208 study that is going to really, I think, transform the hearing field. I'm going to get into more details, but we've done 5 clinical studies to prepare for this particular clinical study. We've learned a lot along the way, and I'll tell you more about what those learnings are and how we've really refined this study to be a success. Second, we're going to be kicking off later on this year a program, sort of phase 2 program in hearing regeneration that I'll give you more detail on. And then finally, our MS program has some of the best preclinical data, I believe the best preclinical data in the remyelination space, and that's going to be in humans in the clinic by the end of 2023. The key takeaway from this slide is this is about a breakthrough way of thinking about healing the body from within, using small molecules and cell signaling to use progenitor cells that are already in the right place to heal the body, again, from within. And again, we have this breakthrough opportunity that I'm going to talk more about in the hearing space. I just summarized sort of the key, 3 factors that I think are really important just to sort of tune into here for a moment. Again, a Q1 readout, a second half of the year readout, both in hearing, and then we're going to be getting into the clinic with our MS program in the second half of 2023. This is all about an unmet clinical need. Right now in hearing, there is no therapeutic solution. There's only mechanical devices, hearing aids, cochlear implants. The ability to create a new paradigm in this area really goes to understanding and having a deep commitment to the underlying biology of the inner ear. And this is about the progenitor cells, the 15,000 hair cells that we're all born with and that really sort of are sprout, if you will, in the 3rd trimester in your mother's womb. And understanding that Mother Nature never, intended for Guns N' Roses concerts or, headphones or, airplanes or subway platforms. And it's these antennas that get damaged or shocked through the various sounds that they're exposed to over the course of somebody's lifetime. That's what the therapy is meant to be able to address. So most of the time when people are looking for a diagnostic solution, there's blood tests or other things along those ways. The standard of care, if you will, is this idea of a 50-word test that patients get. It's a standard test accepted by audiologists and the FDA. And it's really a straightforward test, been validated. It's about hearing 50 words and then responding back what you hear. But the key takeaway is a 5-word improvement is a very significant and is clinically meaningful. And let me give you one example. The blue bar that you're looking at is a cochlear implant patient who was getting 20 out of 50 words correct. However, with 5 additional words, they can delay that cochlear implant surgery and continue on using their hearing apparatus or their hearing aids. And being able to avoid the cochlear implant surgery is a significant clinical, step in validation for our therapy. This is some data that we have. We have more data like this. But if 5-word improvement is significant, we've seen that patients have gotten up to 20-word improvements on these tests. And again, these tests are a patient goes in, works with an audiologist, puts on headphones. There's a random set of words that they're read, and they have to repeat back to the audiologist, and then the audiologist records their response. And with that, and in the way this worked is all these patients got 1 dose or 1 delivery of our FX322 drug into their middle ear. And within 30 days, we started seeing a response, though all of these numbers are based out to 90 days. It's also important to understand that some of these patients maintain improvements for up to 1 to 2 years on a single injection. This data was published in the leading hearing field, Otology, Neuro-Otology, obviously peer-reviewed. It was double-blinded, placebo-controlled. And I like to say, for the first time in human history, we've shown that we can improve a function within the body that never gets better, it only gets worse. And that really laid the groundwork for us doing 4 more additional key clinical studies to understand the totality and the breadth of FX322. And that's what's represented here. Various studies meant that we worked, that we all reviewed with the FDA, that are really meant to understand where are we seeing the best outcomes, where do we want to go fishing, if you will, for our key 208 study that is now, being enrolled. The answer that really, or the path that we really followed, if you use the analogy that my kids use, like Hansel and Gretel and follow the crumbs, it's the idea of the orange dots. Those are all treated ears that got FX322. You can see untreated ears, and then you could also see placebo. And that clearly told us that there's a consistent response that we're able to see. And to take it one step deeper, we were able to see that response in these particular areas. This is what we call, this is where we're sort of gone fishing, if you will. So 33% responder rate for people that had moderately severe hearing loss, sensory noise hearing loss. You also see patients with sudden sensory noise hearing loss responded 27% and 40%. And so as we were designing the current 208 study, that's what we focused on in terms of a patient population. It's also important to recognize that the placebo and untreated ears had a 3% response rate, So as far as the size of that market, you're looking at between 7 and 10 million people in the United States alone. Overall in the United States, the hearing restoration market is about 40 million people. Globally, it's about 1.5 billion people. And it really provides the company a really solid beachhead for us to think about where we want to grow the company and move it ahead from a commercial perspective. So why don't we switch for a moment and think about the current 208 study? What have we done? How have we applied our key learnings? Why do we believe we're confident in the outcome of this study? So first of all, it's going to be approximately 120 patients. It's a 1-to-1 randomization, untreated ears to treated ears. We've instituted a lead-in period, which is a patient comes in, gets their hearing tested, then they come in 2 more times in order to maintain that stability of the patient's hearing. We have the ability to quality control, if you will, all of our clinical studies, all of our audiological studies. So a patient comes in, they're recorded, they're video recorded, so we're really making sure that the audiologist and the patient interaction is of the highest quality. And the sites and the patients are all masked. And so the data, if you will, is put into the computer and decides if the patient is going to get into the study or not into the study. And all of this is designed to be as rigorous as we can to take any bias out of the system. One of the biggest steps forward was really work that we've done with the FDA that led us to have aligned endpoints. About a year and a half ago, there was a patient-focused drug development seminar that the Hearing Loss Association of America put on with the FDA. We were one of the sponsors, where they brought patients in who have hearing loss, and the FDA wanted to hear from them. What's the unmet clinical need? What is it that you want to see, so to speak? And resoundingly, the patients were really looking for clarity of sound. It wasn't so much about loudness of sound. It was, I can hear you, but I can't understand you. And that really laid the groundwork for frequency in the FDA coming to terms of our next clinical endpoints, which is that understanding or speech perception as being a critical endpoint for patients. So as we think about a commercial plan, which we clearly do, couple key factors that as you get to know us, you'll see that will really drive this strategy. It's a small molecule. We're not taking cells out of the body, manipulating them and putting them in. This is all happening in place by targeting progenitor cells in specific cellular pathways. ENTs already do this procedure thousands of times a day around the world. If you have a middle ear infection, even tubes in your ear are akin to this type of procedure. And there's a clear path to reimbursement. There's existing CPT codes, and we fully expect that we'll be able to engage into some meaningful reimbursement discussions. So, a couple key points is that on the regulatory side, we're going to obviously, with the data, apply for breakthrough therapy designation, and we continue to sort of prepare the company to be able to do that. In conjunction with that, we work closely with our partner, Astellas Pharmaceuticals, which is Japan's second largest pharmaceutical company. They have been a longtime partner the past 3 years for us. They paid us $80 million upfront for the rights, for the global rights of Frequency's FX322 therapy, and Frequency retains the US rights exclusively. And we continue to work closely with them. We have about $90 million in milestones upcoming for start of phase 2B work in Europe and in Asia. So just to summarize, we have a lead program readout in Q1 of 2023. We have a second hearing program that will read out in the second half of 23, and we're going to be in the clinic with our MS program, remyelination, a phase 1 study, the second half of 2023. So just one slide that really talks about our second program. This is all about going to school and learning from FX322 and understanding how we get deeper into the cochlea with our therapeutic. And, I applaud Chris and the team for really coming up with a sensible way to think about how to get delivery throughout the cochlea. Right now, we're getting high delivery into the higher frequencies, which is sort of the front door to the cochlea. But this is to get it all the way, down the stairs, if you will, in the cochlea. And the reason why that's important is we believe, or we theorize that a greater coverage will allow for a more efficacious response. And so now I'm going to take you through our remyelination program. A lot of people will ask me, it's kind of awkward. You have a hearing program, you have a remyelination program. Our commitment is really simple. It's to healing the body from within. That's been our focus from day one, 7 years ago when we started this company. We believe you can take hibernating progenitor cells in the body and get them to sort of temporarily regenerate and create this sort of therapeutic solution at the site where it's needed. We started with hearing. Hearing's a bit of a pioneering program, we recognize. However, the MS program is well known. And so as we've gone out with our preclinical data and have talked to industry and talked to leading KOLs, to put it frankly, we're really knocking people's socks off. People are really impressed with our preclinical data. They think that it's very intriguing and they haven't seen anything like it previously. Preclinical is a critical step, but obviously it's not the only step. And so now as we prepare to be able to go into the clinic, we have a lead candidate nominated in our MS program and going through the appropriate testing results that we believe will lead us to the clinic in the second half of 23. So just 2 quick slides. If you look at the blue bar that I'm blocking right there, you can see, where all the other companies, all the other efforts, T3, Lingo, clemastine have been, and you can see where a healthy, myelin is. And so this is the response that's best represented with this picture. And, the frequency single dose induces with new myelin and white gray matter right here. And then you can see how the other known candidates in the field have responded. To give you a little bit of context, these mice were demyelinated for 17 months using the Cooperzone treatment. And then they were gone ahead and they were then, dosed with these different therapeutic options, doing it all at sort of standard dosing levels. And so it was this and more data that's really captured the attention. But the belief is that this company can not only do it in hearing, continue this on in MS, but do it throughout the body. It's a steady approach, both feet on the ground, but really delivering in a new mode of medicine and a new arm of regenerative medicine has really been such a focus for us. So just to sort of wrap up, key readouts coming. So from an investor standpoint, in Q1 and then the second half of next year, there's 2 key hearing readouts. and then we're going to be in the clinic with the remyelination program. We have endpoint alignment with the FDA. And then, we have, I think, a really strong program in remyelination. There's going to be more of that we're going to be talking about early next year. Right now, we have $111 million cash that we've reported, and that gets us well into 2024. And then, of course, we have the milestones from Astellas, $90 million that are pending over the next near term. So I covered that relatively quickly. Maybe before we do questions, Chris and Kevin, anything that you want to make sure that we emphasize that I might have missed or things that we want to cover?
I'll just say, as someone who's been in the hearing field my whole career, these types of outcomes are truly exciting. We've never been able to do this, and we've always theorized it. It always happened in NIH and in labs. We're trying to find this to work out, but recently, to have a company be at the stage was why I changed my career to this group, and I'm really excited about where this is going, truly.
Yeah, we're very excited about the approach of activating progenitors within the body. Has a lot of advantages in regenerative medicine where you're not having to do cell therapy, where you're taking cells out and manufacturing and delivering, and you're not changing the genetic code of the body. So when we think about the safety and the simplicity of this approach, we think there are a lot of advantages. I think one of the other major findings in hearing has been that we've dosed over 200 patients to date with FX322, and we've had a very favorable safety profile with 0 serious adverse events associated with the drug. It's a single local injection, doesn't reach active levels systemically. So a lot of advantages to this approach. We're really looking forward to these readouts.
Kevin, you spent a lot of time putting this slide together. Do you mind just talking through it from your perspective as a trained and skilled audiologist?
Sure.
Please.
So the way we do these tests are always at the same presentation level. So when they start the study is when you establish how loud it's going to be. And so every test thereafter is at that same level. So the only thing you'd expect is the same or worse, because as we hear over time, we get worse. Now, the study's 90-day readout, so you don't expect presbycusis or these things to show up. But when it goes the other direction, that's truly unusual. So these orange dots that are above and quite far above, right? So if this is on a 50-word test, that's changes of over 20 words. And a baseline that could be, 5, 10 words, that's a really big impact on their everyday life. These types of shifts are not seen. Okay, of course you can hear better when you get amplification, but this type of clarity, that's just due to audibility. This is a clarity change. And that is at the same level. That's a really big deal. Now, you could say, well, what's the chance that this is an error? Well, if it was an error, you'd expect to see those on both sides of the graph. You'd have as many going up as going down. And you also see the validation in the untreated and placebo ears. So that untreated ear looks perfect. It's a normal distribution where there's one data point or a couple data points that are in the 2.5% you'd expect to be above the line and a 95% confidence interval. So that's test reliability demonstrated. There aren't as many placebos because all of our previous trials just weren't built to have the equal weighting that we have in 208. So to me, this shows that these trials are behaved well. So those untreated and placebo ears are doing what they should in terms of statistics and probability. And treated does not look like those. And those size of those responses are big. You might criticize that to say, yeah, but look at all those in the middle. But that's where we've learned not to invite them into the 208 trial and how we can enhance that population because these are easily observable in a clinic. And so it'd be quite simple for an audiologist or a surgeon to say, okay, well, pick the subjects more likely to respond. So this to me this kind of gives me goosebumps. We've not seen this type of thing. Those responses are large. And we've learned from these studies to refine that population.
And Kevin, do you mind just talking a moment about this bubble chart that we have and how you think about it?
Sure. So this, just to orient you, the size of the bubbles are the number of people in each of these quadrants. So we tested the fewest people where the bubbles are smallest. And if you go back one, actually, please, the percentages are the number of responders we saw within each. So if we wanted to capture our enriched population, where were the responders? And so we just drew a circle around where people are most likely to respond. The etiologies noise, that can come from a single loud noise event or a history of chronic loud noise. Sudden idiopathic hearing loss is people just wake up one day and one ear often can't hear well. Now, we're very careful to ensure that there's a spontaneous recovery that can happen within a month of that. Often when treated with a steroid by a physician, but we are well far away. We have at least 6 months before they're invited into the study, and they have to demonstrate stability before they get into the trial. So these are the people who had a chance to respond spontaneously but didn't. We waited a long time. So we don't see these types of recoveries in that group. So when you look at that group, that's who we invited to 208, and it's still a lot of people. And if you blur those edges a little bit, that number doubles. So it's a big market, and it's a real opportunity to help a lot of people in need.
Chris?
Yeah, and then if you turn to the MS green slide, the last point I'd make is right now there's all the therapies in MS slow down the attack of the body on the myelin. So the immunomodulators, very successful, $20+ billion a year in sales, but nothing rebuilds myelin, and that has been the holy grail in the field. There have been multiple attempts, as you've seen here. Some of these advanced to the clinic. As you can see, we believe they have pretty modest efficacy preclinically and have had pretty modest efficacy clinically. So we at Frequency discovered a new target and have designed NCE's new agents that hit this target that we don't believe anyone else is going after, and believe it's just a major step forward in terms of the power of the approach. So we're really excited about getting this in the clinic as well.
One question then we'll open up to the audience, Chris, for you is how do we think about it from why is MS and hearing connected? You know, what is it that's the underlying strategic scientific commitment that we have?
I mean, our expertise is how to control progenitor cells in the body, how to get them to activate, grow, divide, and become the types of cells that can repair tissue. So there are progenitors in the ear of one type, and it's a distinct type of progenitor in the brain. So we communicate to them with different molecules that approach different pathways, but the fundamentals are the same. The drug discovery approach is the same.
That's great. So why don't we open up for questions? Unless we did such a good job that there aren't any.
So are you also looking at different types of hearing loss and how that might be treated differently? And is there a genetic component to this as well?
So the way I think of genetic hearing loss could be that it's like the area is not conducive to supporting the hearing organ, the organ of Corti. So we are avoiding genetic hearing losses by going after adult onset. Most genetic hearing losses occur at childhood at birth. Because we just don't understand the mechanism as well. Often in these cases where let's say you don't have an endocochlear potential, you don't have other crucial building blocks. We're taking cochleas that have heard well and stopped hearing well. And that is the largest group. So we're absolutely looking at etiologies. There's other etiologies that would never respond. Say you had a transverse fracture of the temporal bone and broke the blood supply. There's nothing you can do from a progenitor cell activation approach. So absolutely. We're looking at those cases where you have a good infrastructure that was damaged, where we can put it back together.
Well said. Yeah.
Thank you. Maybe if you could go to slide 10 again. I understand the rationale of choosing the middle swath of patients here, but could you speak to maybe the mechanism that you're testing and its potential applicability beyond the 208 patient population? Would you expect to see an effect that just maybe may take a larger study, or how would you think about that moving forward as well?
Yeah. Oh, go ahead.
Oh, sorry. I thought you were looking at me to respond. First, let me talk about the test. So the test has a psychometric function where if you have really good hearing, you cluster at the top. There's not a lot of room to get better. And if you're really poor hearing, you're on the other side. So there is a possibility that the test we're looking at is really responsive for these subjects in the middle. And one of the things we may do later on is to expand that by using different types of tests. So we don't have a deep bench when it comes to speech perception tests. So the ability to get at more people by using tests better designed for them could be useful. So that's not mechanistic. That's more on the detection side. But I'll let Chris speak to mechanism.
That's in addition to that point, which is right on. And patients often start mild and kind of move through this. And that's not thought to be a qualitative difference in what the underlying pathology is. So I think as we look into these different populations, look into additional testing, which can be more sensitive to different ranges, we think there's real potential.
Great. Thank you so much. And then a follow-up for Chris maybe. I was hoping you could just compare and contrast the advantages of your approach versus some of the gene therapy approaches.
Yeah.
I know you touched on this earlier, but maybe you could dive a little more deeply into your small molecule approach to kind of reprogram these progenitor cells.
Yeah, exactly. I think like with the question earlier, genetic hearing loss is a very narrow population where there is something genetically wrong with the way the cochlea is built, and groups are trying to find specific mutations. They can go in and try to repair in a timely fashion to fix that. There involves gene delivery, right? Those are large packages you have to try to get into the cochlea, which is this tiny organ sealed in the hardest bone in the body. So our approach has a number of real distinctions there. We're going after the very broad population of people with acquired hearing loss. It's the overwhelming majority. 80+% is sensory neural hearing loss. Also, we're doing this simple intratympanic injection that's used for steroids and antibiotics all the time, because with a small molecule, you just need to place it in the middle ear, right behind the eardrum, and it can diffuse into the cochlea. There are membranes that let small molecules in. Not possible with something like a gene therapy. So bigger population, simpler delivery is really the distinction.
And let me speak a moment about the clinical trial for that. Most of my clinical career was working with babies who get cochlear implants. So the standard of care is for a baby to get 2 cochlear implants. And with that, they learn language and music and dance, and that's great. Think about the trial design, right? So they're gonna have to detect this gene very early, and then only give them 1 implant and the gene treatment, and then wait years to determine if that difference is subtly different from 2 cochlear implants, which would be the comparator. I think that's a real tricky path to approval, given the standard of care and given the efficacy of that. So I applaud the effort. It's important. These devices are wonderful devices, but they're a lot to maintain. So I think it's a hard path. I truly don't understand how they're going to be able to get through that, despite all the great efforts that they're making.
Great. And then one of the mentions was about the ease of administration of these small molecules and that they can diffuse across the membranes in the ear. Maybe you could talk a little bit about the work you've done to ensure you're getting good drug exposure throughout the ear. How confident can we be that you're getting this exposure throughout the cochlea with this transtympanic injection?
Yeah, so I think we're the only group that's ever done a study that's confirmed we have drug entry to the ear for a hearing restoration drug. So we did a human study where patients were going to be getting a cochlear implant, and before their implant, we gave them FX322, and that's a time when they get their implant that the cochlea is opened, and they could go in and sample the fluid from in the cochlea and see that in fact we're getting both drugs, and based on the modeling and all the preclinical work we did, they should be in the effective targeted ranges, largely in the high frequencies, which is the cochlea is just like a piano with the high frequencies at the entry point where the drug goes in. But with 345, the second program, what we saw is, what if we could cover more of the cochlea? So we did that by developing a more potent compound, so you need far less drug to be in the therapeutically effective level. So with that, we've confirmed preclinically that we have reached that effective level much further through the cochlea, and we can model that over to humans as well. So we're really excited about moving that ahead.
That's great. We keep talking about these devices and hearing. It seems like it's the only thing we really have to benefit patients with hearing. There's been recent legislature that passed around hearing aids. I was wondering if the company had any comments around the greater accessibility of hearing aids and potential impact that could have on the commercial opportunity for your therapies.
Yeah, Kevin, I'll let Kevin comment.
Yeah, sure. I was involved in a lot of the steps of the creation of the bill, the law, and the implementation. So anything that gets people thinking about their hearing is going to be good for us. I think it's Sony announced today they're entering the field. So if people can engage in their hearing health earlier, we feel very confident they're going to get to that point where they're going to say, it's louder, but I still need it clearer. And there's nothing to say that you can't use both a hearing device and a hearing therapeutic together. And in fact, that could be synergy to the referral pattern. So someone who fits a hearing aid says, great, it's louder, I hear more things, but I'd also like it clearer. Great, go see my physician friend, or my physician friend comes on Injection Tuesdays and gets it all done. So I think there's synergy, and I think anything that gets people thinking about hearing is good for all the different solutions that are out there.
And I should just note, Kevin is the chairman of the Hearing Loss Association of America, which is a not-for-profit, but the largest patient advocacy group in the space. So we're very fortunate to have him from that leadership perspective and as a well-regarded audiologist.
Thanks, David.
Thank you.
Great. I don't mean to monopolize the mic here, but maybe in MS, could you just speak to how quickly we could get proof of concept data from that study? I know it's really interesting preclinical data, and the IND is expected next year. How early could we see really positive human proof of concept?
I'd love it as early as possible, but I'll let Chris answer that.
I think we've been able to benefit, we'll be able to benefit from the fact that others have taken remyelination agents into the clinic. And there's been publications showing benefit to patients, for instance, with MS with optic neuritis. So there's a lot of things we can learn from Biogen's experience, from the work with clemastine and some other drugs that will inform us about how to generate that information. But as the program progresses, we can give more on the timelines.
Thank you. We really appreciate the opportunity to speak. Thank you for coming, and we'll be around if you have any other questions. Thanks.
Thank you.